Hi friends, let's study about cell physiology. I am going to discuss volume measurement and disturbances of body fluid compartments. Let's start with volume. So, total body water. In our body, the total body water is 60% of the body weight. Let's consider a 70 kilogram healthy person. So, in a 70 kilogram person, the total body water would be 0.6 into 70, his body weight, is equal to 42 liters. So out of this 42 liters, 2/3 is intracellular fluid and 1/3 is extracellular fluid. That is 0.4 into his body weight, 28 liters of intracellular fluid, and 0.2 into his body weight, 14 liters of extracellular fluid. So out of this 14 liters of extracellular fluid, 3/4 is the interstitial fluid, roughly about 11 liters, and 1/4 is the plasma, and roughly about 3 liters. So total body water is 60% of the body weight, and total blood volume is 8% of the body weight. So in a 70 kilogram healthy person, 0.08 into 70, so roughly about 5.6 liters of blood. Let's consider 5 liters of blood in a person. Out of 5 liters of blood, 3 liters would be plasma and 2 liters would be cells. So 8% in 8% of the body weight, 5% would be plasma and 3% would be cells. Let's discuss the measurement of body fluid compartments. So there was the question asked, radiolabel albumin was used to measure the options are total body water or ICF or ACF or plasma. So the answer is plasma. We will see in some time. So what is the method used to measure? Dye dilution method. I is the initial volume of the dye injected into the body, and C is the concentration of the dye in that particular compartment, and A is the volume lost out of the body. So this is the equation. V is equal to I upon C, and in some cases, if some dye is lost out of the body, the final equation would be V is equal to I minus A divided by C. So this is the question. They given I is equal to 10 grams and A is 1 gram and concentration is 50 milligram percent. So let's calculate 10 minus 1 grams divided by 50 milligram percent, and let's convert everything per liter. So here 9 gram per liter, and milligram percent is milligram per 100 ml. So it comes like 9000 milligram divided by 500 milligram is equal to 18 liters of extracellular fluid, and we know that 3/4 is interstitial fluid and 1/4 is plasma. So what are the dye we use to measure the body fluids? So to measure total body water, the dye used is deuterium oxide, tritium, antipyrine, and aminopyrine. So these dyes are injected into our body. The dye spread all over the body. We can measure the total body water. And what if you want to measure extracellular fluid? So the dye injected should stay in that extracellular compartment, and it should not go inside the cells. So the dye should be non-metabolizable saccharides, and one of the most frequently question asked is inulin. You should not confuse with insulin. So inulin is a fructopolysaccharide. It doesn't metabolize into cells and it hardly enters the cell. Okay, so then mannitol and sucrose also used. So deuterium, if you are given deuterium space, that is total body water, and if you are given sucrose space, that is extracellular fluid. So simply to measure intracellular fluid, automatically ICF is equal to total body water minus extracellular fluid, otherwise called as deuterium space minus sucrose space. To measure plasma volume, so I asked this in the starting. How will you measure plasma? We can use radiolabeled iodine I131 or radiolabeled albumin. So to measure red cell volume, you can use chromium 51 tagging. So let's see clinically. So what are the disturbances? What are the disturbances happened in the fluid compartments? What if the disturbance happened? So if there is some disturbance in the fluid compartments, there will be dehydration or overhydration. Dehydration is water lost from the extracellular compartment, and overhydration is water gained in extracellular compartment. So there are three types, and first one is isotonic dehydration. In isotonic dehydration, let's see in this diagram. This is intracellular fluid compartment and this is extracellular fluid compartment, and the osmolarity is, the normal osmolarity is 300 milliosmol per kilogram. In isotonic dehydration, the tonicity remains the same, that is the 300 milliosmol per kilogram is normal and there is no changes in the tonicity, and these lines are the disturbances happen in these compartments. So water, in isotonic dehydration, water and sodium lost in equal proportion. So here you can see water and sodium lost in equal proportion. So there is no changes in the intracellular fluid compartment, only water lost in the extracellular fluid compartment. So this happens in GI fluid loss, burns and hemorrhage. Let's see about hypertonic dehydration. In this picture you can see this is the ICF compartment and ECF compartment, and the tonicity normal is 300 milliosmol per kilogram, but here the tonicity is high, right? What happens? Only water lost from ECF. Here water and sodium lost in equal proportion, and but here only water lost. So water lost means sodium is high, water is lost and sodium is high, the tonicity is high, right? So sodium, what the sodium will do? Sodium loves water, sodium pulls water. So here the sodium pulls water from the ICF, so the ICF volume shrunken secondarily. So this type of cases is seen in diabetes mellitus, diabetes insipidus, alcoholism and injection of lithium salts. Then hypotonic dehydration. Here in this picture you can see the tonicity is low and the ECF extends. So what happens is here water lost, but sodium loss is much more than water loss. So here sodium loss is much more than water loss. So sodium, there is some water but there is no sodium. So here sodium in the intracellular fluid pulls water from here. So the intracellular fluid volume increases secondarily, so the cell bulges. Tonicity is low and the cell bulges. It happens in primary hypoaldosteronism and primary hypoadrenocorticolism.